Hi everybody, I would like to welcome everyone to today's webinar. It is 5 p.m. in Europe and 11 o'clock on the east coast of the US. We're getting started on time. Today is entitled PDX, Predictive Cancer Models for Better Precision Medicine, which is being presented by Jonathan A. Wing. Before we start, I would like to introduce myself. My name is Krista Vitrouille, and I am a business coordinator at Oncodesign, and I will be your host for today's webinar. First, I'd like to address a few practical matters. The webinar will be available to all attendees. We'll mail you a link to this presentation by email. After the presentation, our presenter, Jonathan E. Wing, answer a few of your questions. So if you have any during the presentation, please type them in the control panel that you can see on your screen. Your attendee won't be able to see your question if you send them directly to the host. So that will be me. If you cannot see the control panel, just click on the top arrow of the top of your screen. In case there are questions that cannot be answered by the end of today's session, data will be responding to you by email within a couple of days. Now, if you experience technical difficulties, please message to the host and someone will be able to assist you. Introduce Jonathan E. Wing, our speaker today. In 2000, Jonathan joined Oncodesign, where he has served in various business development roles until becoming the company's business director in 2008. Prior to Oncodesign, Jonathan was involved in research at the Roosevelt Park Center Institute and Millennium Pharmacology. He holds a degree in molecular biology and molecular genetics from North Dakota University, and an MBA in life sciences from the Open University in the UK. Let's begin the presentation. Just will guide you through the slide and that you show you today. Jonathan, you ready to start? It's like we are not hearing you, Jonathan. Do you have your microphone on? It seems like we are encountering some difficulties. Let me see if we can fix the line with Jonathan so that he can start the presentation.
Well, I apologize for that little technical problem, and Krista, thank you very much for the introduction. As she mentioned, today's webinar will be on PDX and having better predictive cancer models for precision medicine. A little bit of introduction about Oncodesign. We are specialists in the area of oncology pharmacology. We are based in France, and we've been working in this area for about 20 years. We do everything from analysis, cell models, PDX models, humanized for the immune system, and pharmacoimaging, all the way through the discovery up to preclinical toxicology. So what I'll do to begin is just give a little bit of rationale behind the need for some of these models. Obviously, in oncology, a lot of the drugs that we have are good and they're very interesting, but we still are not able to give long-term survival to 60 to 70% of the patients who have these drugs that we give to them. Personalized medicine is still ongoing, a lot of targeted therapeutics and immuno-oncology underway, and there are still a need for new drugs for very, very specific subpopulations of patients, including resistance. Other problems that we have are some of the high attrition rates in drug development in phase 2 and in phase 3. Also have a high cost of drug development, and even though we invest more and more money in the development all the time, we're not seeing better success rates, and we're not seeing advances early through the clinical development and getting drugs approved. We also see issues with clinical predictive power of the current models and technologies that we use today. So the need that we're looking at from our standpoint is having highly characterized models that are representative of the high variability of patient tumors, to have more clinically predictive models, and to identify and to associate very specific biomarkers with those models as well. So PDX is one area of many. It's a very, very interesting area in terms of trying to make a better prediction of clinical outcome very early, taking into account the diversity of a phenotype and a genotype of a patient and representing that within the model. Identify biomarkers of response or companion biomarkers to help us to stratify patients very early in the clinical development process. And then estimated that by a group of PDX, we can actually reduce the cost of preclinical and clinical development by $80 million in about 2 years in terms of the timelines. So the first thing that we will look at when we're developing these models is to start with the clinical need and then move those into in vivo models that represent the patients themselves and have as much information and characterization as possible. And I'm moving into some more proof of concept and preclinical phase 2 trials as we go along. The idea is to take those compounds through into clinical development, into phase 1, 2, and then into phase 3. And obviously, as we have some of the early clinical trial results, we will see drug resistance, and that is actually one of the areas of most unmet need, is to start to look at having resistant models for very, very specific compounds that have been newly approved. Areas that we're going to begin to look at much more in depth over the next few years. So as we develop these models, we want to associate in vitro cell lines that go with those, DNA and RNA banks, protein banks, serum banks, and stool samples in the biobank that we can go through and screen and to take a look at early screening stages for some compounds, and then have everything from the in vitro screen, the in vivo and ex vivo assays all the way through early clinical trials into a common database that we can associate all of the data and help us to make predictions as the compounds advance through the clinical development. Service provider, we will look at what might be available to the sponsor. How can we help you as a user as much as possible? And the first thing that we will do is develop the models and be able to let you use them in-house. So if you're interested in acquiring models, collaborative work to share models, all of that can be done, and we can provide the models for you to use in-house. Or we'll take the molecules and then use those at our facility, do the drug evaluation here, and generate data, results, conclusions, and a direction forward for you as well. And so that's a twofold type of business model that we have in terms of getting you to do your research. So in terms of the clinical value of these models, you can get a better evaluation of targeted therapeutics compared to standard xenograft cell lines. target identification and validation using fresh clinical and preclinical biological samples is much more interesting to do. And then from that, build models with better histological and molecular characteristics. The idea there is to validate molecular targets in vivo and to evaluate the activity with respect to the patient profile. One is with the panel of colorectal models. It's one of the first things that we looked at into developing was compare with some of the clinical results. So the diagram on the left is a comparison in clinical trials of patients who have mutated KRAS versus patients who have wild-type KRAS and that have been treated with cetuximab. And the wild-type patients do have a little bit better response. And we've been able to mimic that same type of approach using our models from the model that we developed in PDX, looking at cetuximab as well. So the first effort that we went into very in-depth model development and characterization was done through a consortium that was called CRAC. It involved a number of French pharmaceutical companies, a few of the smaller biotech companies, and different areas of expertise, including Oncodesign. And also being able to put in place a number of different academic groups as well, from which we got not only material, but also able to do the clinical trials afterwards. So that was a very, very interesting collaborative work we did to generate some fruit from that. The first panel that we did was in colorectal, obviously. It was one of the easier ones to build. It was good proof of concept. We collected 86 tumors, most of those from primary site. We also collected from hepatic metastases and some other metastatic sites as well. And from there, generated 53 models, mimicking that ratio, having mostly primary tumor, but also having a number of models that come from different metastatic sites. And we actually, in the panel that we have 8 matched sets of metastatic and primary tumor models that came from the same patient. So it actually is an interesting comparison data that we can do with those models. So in terms of histology, from the sample, the original one, we can look at well-differentiated, well to moderate differentiated, or poor to differentiated, and after passaging those in vivo, we can maintain the same histological subtype. And obviously that is in direct contrast to the type of histology and the undifferentiated status of all the cell lines that we look at. So that's one of the key differentiating factors with these models that makes them much, much more interesting for further development. We did gene mutation frequency comparison along our model for actionable hotspots and gene spots that are common in colorectal cancer. The models that we have, the 53 models, is a percentage of P53, APC, KRAS about half the time, and that mimics a vast majority of the cosmic data that's out there on the clinical samples. So again, we have good correlation with our panel as to a lot of the clinical situations that we have as well. In terms of gene expression profiling, we actually will do profiling of the original tumor sample from the patient and the graft, the first round of grafting. And there we see the gene expression differential and profile here. As we go from early passage to late passage, we see very good correlation and good correlation between mice in the same passage. And really the only difference that we saw in the two differences at the beginning was due to stroma. And the mouse stroma overtakes the human stroma relatively quickly for passage, and that's where the difference in the genes that we saw at that time in these rounds. We also did a lot of pharmacological data. We generated testing of 5-FU, oxaliplatin, CPT11, and cetuximab in all of our models. And what we can do at this point is get, for each model, idea on the mutation status of the hotspots. We have full transcriptome data that we can do for the gene expression profiling. And then we'll compare that with drug efficacy data and response to various chemotherapeutics. And we actually then look at correlation of something such as KRAS wild type and mutated, resistant or sensitive to cetuximab, and then using those as patient subpopulation sets and doing some comparison work with novel compounds or different inhibitors in combination and looking how those might change and run that as a preclinical phase 2 trial. It's a very comprehensive panel, a lot of information there as well. What we've seen is when we segregate out of our panel, half of the models are mutated for KRAS and the other half are wild. We definitely see the same thing in the clinic, mutated, not very good response at all, and the wild type models, a rather good response in terms of overall survival. So it's one more correlative fact that these can be interesting models to use for phase 2 clinical studies. And there, the next step is to look at, PDX models are wonderful for a lot of things. We've got mutational status and heterogeneity that's very, very close to what goes on in the clinic, wide variety of histological types, really interesting models to be able to do that, and they're commonly used in those areas. However, there are areas of improvement. There are not many models that are metastatic, very few of the PDX models that are available are orthotopic, and there are almost none that are used in a humanized situation where we may have a humanized immune system, humanized liver, humanized skin, those kind of things. So those are areas that we are focusing on and working on as we develop our PDX models to go down that road of better grafting, microenvironment, and better humanization. So the examples that I will show here is some of the metastases that we've observed from primary orthotopic graft. We grafted all of our models, 53 models, we've grafted all of them orthotopically. None of those from a subcutaneous graft yield liver metastases, but 15% of those models will yield to a certain extent liver metastases, and sometimes up to 40% of the time. So MRI, quite commonly, to look at the location and size of the primary tumor. That's what we're seeing here. And then we'll also use MRI in the liver to look at different metastatic lesions. And we can use MRI to monitor the onset of when those lesions appear, when they begin to disappear after treatment, how much inhibition we may have at that point as well. And the diagram here shows treatment with CPT-11 versus untreated. And when we do treat with CPT-11, no metastases whatsoever. And when we have a control group, roughly 5 of the 12 animals had metastatic lesions in the liver. So it's a great model system, and it's a great tool that we can use with imaging to monitor what's going on in the area of metastases and orthotopic primary tumor growth. So this happens all the time. If we were in location that we need to be, we should be seeing metastases quite often. It's a clinical situation, quite often metastatic lesions. Why aren't we getting that? And one of the hypotheses that we have is that it's linked to the human versus murine stroma. And so that's the area that we want to look at next to humanize. So we've worked for over 10 years in the area of humanization of the immune system. We've worked with NSG mice, NOG mice, second-generation NOG mice. We're looking now at other species as well, other strains of mice, to be able to do that more effectively. We've humanized liver, we've humanized for skin, which is very interesting in an EGFR situation. And now we're starting to look at tumor stromal, specifically in the area of breast cancer right now, and taking our PDX models and humanizing those with mesenchymal stem cells to get better microenvironment. And that's underway right now. What we hope to do with this is to increase the relevance of our models with respect to what's going on in man, increase the incidence of metastatic disease, which is rare in PDX and very systematic in patients, and to have better prediction of the efficacy and potential side effects of drugs that are under development. So we'll go models at that point. Of course, having in vivo systems, in vivo models is a great link to going into the clinic. However, to do that, we need to maintain these models all the time in vivo. We need very comprehensive animal facilities. We need very specialized technical staff and human resources on that side. Also, there's a long period of time, very time-consuming to have these done, and if you want to do anything on demand, they constantly have to be grafted. So, large number of models, large number of animals being used all the time. Now, to overcome these disadvantages, one way that we're looking at is to look at ex vivo assays, Collier's two-dimensional, three-dimensional assays to give better predictability and better screening of compounds before we go in vivo, or looking at developing mouse clinical trials with very, very small N numbers and a large panel of PDX models. And that's another area that we want to do for screening activities. So what I'll do as part of the presentation go through about 3 different assays that we have been using routinely. The first one that we started with was basically just taking a tumor fragment, resecting it from the mouse, putting culture, drug, and then embedding them into a array. After that, doing IHC for Ki-67 or caspase-3 for apoptosis, and looking at a chemosensitivity profile with those drugs. And example we have is one of our PDX models. We did that assay. We ran it with oxaliplatin SN38, which is the active ingredient for CPT11 5U, and we saw of the 3 compounds in this assay system, SN30 had the most effect. It was basically the active ingredient of those 3, which is a common cocktail in clinical trials. In vivo, we saw very similar correlation. Oxaliplatin inactive, 5FU not active either. The CPT11 did show some activity, and so we did a very good correlation there between the ex vivo and the in vivo situation. However, it's still IHC, it's time-consuming, and it usually requires specialized facilities to be able to do that as well. Assays that we are now looking at, and we had some publications in 2013 on the use of tumor spheres, or very specifically spheres, for a colorectal panel. Very similar to what we would do with the cell line, in vitro culture, and develop spheroids, and then do drug assays. We have 2 options. We can take the patient material directly and then associate that into culture, or we'll take a tumor resected from a PDX and then also dissociate that and put that into culture. Get tumor spheres, close up, and then treat with drug and medium, and then do viability and cytotoxicity assays. It's relatively standard at this point. So the first thing that we found interesting is that in a colosphere, even after 17 days, these cells do remain viable, they proliferate, they're very, very active. We also saw that the layers, there's not much communication and there's not much apoptotic activity after treatment with drug. When we do have a colosphere, the communication remains intact, and that contact between the cells has a certain memory. So even if we treat with ETA or anti-E-cadherin, we still see apoptotic activity after treatment with drug. And the other thing that we found interesting is when we take those colospheres and we inject them into mice, usually that they take. So the first that we did was put them into a highly vascularized situation in the kidney. The colospheres 2/3 of the time would take into primary tumor. About 1/3 of the time with spheroids from cell lines, that would happen. And then we looked at lung metastases from those primary grafts. And 6 out of the 15 total and 6 out of 10 of the colospheres took would actually yield metastases in the lung, whereas the spheroids would not at all. So it's that a situation, if we want to look at other ways of looking at metastases and effect on metastases, colospheres could also be a very interesting in vivo technique to be able to do that too. So there, just an example again with colosphere, 2 different models, which in vivo we know is to be sensitive to CPT11, and in vivo which we know is to be resistant, the 2 models here. And then using the colosphere assay, we see correlative data, active ex vivo in the same model that showed in vivo response, and not active in the situation of the model that had no in vivo response as well. So good correlation with that test as well. And through a consortium that we have in place today, we are collaborating with a company called Oncodesign, which actually has a little bit of a faster type of assay, and a little bit easier to use, which is 2D. Very similar though, we'll take a patient tumor or a PDX tumor, dissociate those, put those into primary culture in a 2D culture, and that's used with very specific media that Oncodesign has developed, and run cytotoxicity and proliferation assays using microscopy. And it's quantitative, we can count the number of cells and look at drug response in this assay very, very easily. It's about a 2-week total time from dissociation to results, but it works pretty well. Now, one thing that they looked at during the evaluation process was to take retrospective studies of patients. So they looked at different patients and took samples from them after treatment, and they went into looking at different agents, single agent, drug combinations, and profiling those. And the first patient is someone who didn't respond, and we didn't see much activity in the assay either. And at patient 3 and 4, actually pretty good response to the combination, less to some of the signal agents, and there was very, very good correlation between these results and what had happened in the clinic. So it gives us a lot of interest that this assay is going to be a very, very good screening tool for us to be able to run through drugs at that point. So what we want to do with this test is to be able to validate some of the molecular, proteomic, and biomarker diagnosis with a standardized and highly functional assay. Obviously, we choose the most effective drug to treat a patient in a prospective manner, but then we'll look at drug combinations or looking at new drugs in this assay as well, and maybe predicting whether or not a novel compound might have some hope, give some hope for that patient in terms of response. So as we get a little bit near to the end of the presentation, I just want to give an overview on the models that we have. I spent the entire presentation talking about colorectal because we had a lot of depth within that model. We've done a lot with those models. We've done a lot of things with them. They're very, very well characterized. We now have a panel of 20 established models for breast and another 20 that will be on the way that we should have by next year. There are partly triple negative, but also some HER2 positive or HER2 low. Very interesting. Lung models as well. It's about half non-small cell, half small cell, and we've got another 30 models on the way, specifically looking for EGR mutations that we want to introduce there. And then continuing to build our panel out over the next 2 or 3 years in terms of ovarian, kidney, prostate, liver, pancreas, etc. And the way we're doing that is actually through a consortium called EMOD. It's a French initiative with a number of different pharmaceutical and biotech companies dedicated to developing and characterizing these models. Oncodesign will be the sole service provider using these. The cancer pathologies that we want to look at will be very specific, based on clinical and pharmaceutical needs, with a standardized process, and pooling all of those resources together, and so we can share data, share the cost burden, and to put all that together in the same manner. So the consortium partners are about 5 of them from the pharmaceutical industry, 6 from different small companies, 7 academic institutes that represent 13 different clinical centers and research labs. That's where most of the material is coming from, and then eventually clinical trials can be run with them. It's about a $50 million budget, and there's a 7-year program over time. And just to close the presentation, talking about what we will do in the characterization of these, we're into the first year of it right now. We will be collecting from over 800 patients, tumor, blood, stool samples, and all of the clinical history, obviously with patient consent, et cetera. Go through our model development, and I highlighted in red the fact that we are going to look very specifically at orthotopic grafting and also developing cancer stem cell lines that can be used for stem cell approaches and also some in vitro screening, and then having at least 200 models with associated blood samples and associated stool samples. The stool samples are interesting because we want to look at some gut microbiota. So when we do have all the characterization, we have histology, CG arrays, genome sequencing data, RNA-seq, and pharmacology of at least 4 different drugs, and then also having a gut microbiota characterization to go with that, which once we start to do data mining and look very, very in depth, we can do biomarker characterization, identification in association with drug efficacy, prediction of response, and really trying to monitor the efficacy of the treatment, predict how this might go into a patient situation, and also monitor as we go through with some of these biomarkers. So that's what we're going to be doing over the next few years, focusing on very specific areas, as much characterization as possible, and also humanization as much as possible as we go through this. So thank you very much for your time. I appreciate that, and I think we might have some time, Krista, for some questions.
Yeah, thank you, John, and we apologize to everybody for the technical difficulties at the beginning of the presentation. I hope that today's audience appreciated the webinar. So then, there were a couple of questions that came up during your presentation. I want to use the next 5 minutes to address some of these. The question was, to perform imaging modalities other than MRI?
So definitely, Oncodesign has PET capabilities. We do FDG PET, but also other kinds of PET. We radio tracer programs of our own with novel radio tracers. We do SPECT, CT, and we also can do some optical imaging. So if we want to look at different modalities, we want to look at biodistribution studies or microenvironment in terms of angiogenesis, hypoxia, those kind of things, we can definitely look at that in above just quantifying the primary tumor and the liver or different metastases. We definitely have things that we can offer in that area.
Thanks, John. There's one question that I'd like to raise here. I'm interested in providing your models for other consortia than MOD.
Definitely. This is a very European focus that we have. I know that there are pharma-focused consortia as well in the U.S. Both Merck and Lilly have a very strong focus in this area in consortia. We're looking to become part of that. We're also looking for other worldwide academia and other areas to belong to those consortia. I think the idea for us is that models become more publicly available to everyone, sort of in PDX ATCC, if you will, and to make sure everybody has the necessary models that they need to do the best screening possible in the preclinical environment.
All right. Before getting to the last questions, I'd like to apologize to anyone whose question could not be answered, and there's quite a few. We'll get back to those whose question were not answered today via email. Let's have two more, and then we'll close the presentation. One we can address is a question that is: are the patients you obtain the tumors from dry?
Most of the time, it'll depend on the indication. So for the colorectal panel, nearly all of the patients had been treated prior, so we knew which ones were resistant, we knew which ones were sensitive to certain drugs. The memory ones, on the other hand, are mostly naive right now. So what we're looking at is trying to find some more that are hormone resistant, resistant to Taxol, things like that. So it will depend on the tumor indication that we're looking at. It's a little bit of each. The models again mixed. They're a little bit of both. So it will depend on the indication, the source of the material that we got.
All right, let's go for one last question. Do you have any of your PDX in nude rats?
Right, so I think people attending this webinar might know Oncodesign as well for this type of area. We've been working in the area of extended pharmacology using nude rats. It's an exceptional pharmacological tool for us. And the colorectal panel, we've engrafted all of those models into all of the tumors into nude rats, and we've got of those about 35 that take very well in nude rats, and that allows us to do stage pharmacology, continuous infusion studies, sometimes better imaging, and it's an excellent late-stage preclinical tool before going into preclinical toxicology studies.
Okay, well, thank you, Jan. I want also to thank everybody for attending this webinar today, and we wish everyone a very good day. Bye-bye.